That's not good. So stem cells are undifferentiated cells. What that means is, as you know, we in our body we have thousands of different kinds of cells. We have some major classes like neurons or muscle cells, pancreatic cells, liver cells. Within the liver we have liver duct cells, we have other kinds of cells. In the heart, these are muscle cells, but they're a special kind of smooth muscle cell. So those are all differentiated. A stem cell is undifferentiated, which means it can become all sorts of different kinds of cells. So this stem cell here, on the left, this is red blood cells, white blood cells, part of the immune system, muscle cells, smooth muscle, as well as cardiomyocytes, which is for the heart. We have all kinds of neural cells. We've got a beta signaling cell, we've got structural cells, all kinds of different cells. A stem cell, again, is undifferentiated. Or, now there's different kinds of stem cells. Some stem cells can become literally any kind of tissue, and others can only become limited types of related tissue. So, in the bone marrow, those are adult stem cells that become blood cells of various types, including red blood cells and white blood cells. So, now, during normal development, they can give rise to all sorts of kinds of cells. As adult stem cells, they continually replenish dead or worn out cells in most parts of the body. And then, as I mentioned, most adult stem cells can give rise to a limited number of cell types. So here we have okay, so the let's see. These are differentiating stem cells, okay? This is the dermis, epidermis. And as they propagate out, they become terminal differentiation, which is, that means they get to be adult stage and they are fully mature cells. Okay. And stem cells, they can be used. Now, when they were first discovered, there was a lot of thought about how they could be used therapeutically for treatment. So, adult stem cells, in elderly mice, they've reversed those they've used adult stem cells to reverse muscle atrophy. And a number of different things, but they didn't really, it looked like there was more potential than has actually been accomplished. First of all, because these are undifferentiated stem cells undergoing replication, they can get mutations, and that can lead to cancer. So some of the first uses of stem cells had some pretty unfortunate consequences because of that. Recently, they've been able to turn skin cells into more like embryonic stem cells. So they're skin cell, adult stem cells for skin can be turned back so that it can be differentiated into much more than just skin cells. And so, obviously, wound healing involves tissue regeneration. But, of course, it can only go so far. Now, these are planaria. These are multicellular. But if you chop these guys up, chop, 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 and then decide, I really don't want them for dinner, you can keep them in medium, growth medium, and they'll regenerate into complete planaria that will be a clone of the originals. So, actually, okay. So, let's see, we go, this is zone of fission, right in here, where the stem cells kind of regrow. And then there's new asexual regrowth there, sexual regrowth there. Okay. Unfortunately, we can't do that. We can't go with humans. Oh, yeah, sorry.
Oh, no, you're fine. I have a quick question. For, like, reptiles with snakes, like, they release their tail. When they're doing that, do they go through some type of pain? Do you know?
Oh, I imagine it's probably painful.
Okay.
But a lot of lizards, of course, lose their tail.
Good morning.
So that something that's trying to eat them goes after the tail while they escape, right? The thing is, that's designed that way for that very reason, because the tail keeps, you know, moving around and all that. Okay, so I don't know how much pain there is. It's probably not like if we cut off one of our arms. Okay, those are regenerated from stem cells, the whole tail. The planaria, the whole planaria is regenerated from stem cells. Okay, so if you chop that up, you'll end up with the whole planaria. Starfish is the same way.
Are they able to do it as many times, or is it just that one time? That's it?
I think it probably is just one time, I would imagine, for each one. Now, there's other things. You've probably heard that salamanders can regenerate limbs and things like that. We'll look at that in a second. If you actually think about it, though, like plants can regenerate all the time. They have a much more compartmentalized architecture that allows them to kind of regenerate in a way that animal cells just can't do it. Okay. Now, newts, these are types of salamanders. can regenerate limbs. You can actually cut them right off, and they'll regenerate. It takes a while. So, of course, it would be great if we could regenerate limbs. It'd also be great if we could, someone who's had a spinal cord injury, we could use stem cells to bridge the gap in terms of the spinal cord where it's been cut and restore function. They're getting closer, but it's still quite a long way to go, I think. Obviously, yeah.
So, the stem cell that they were talking about in the
Salamander.
no. What's it called? In the what is it? The womb or whatever.
The planaria?
Yeah, there you go.
Yeah.
So when they study that, how do they get that? Do they study on like people who have abortions or?
Oh, how do they get embryos?
Yeah.
Oh, okay. So they get IVF embryos that haven't been used, let's say. Could get them from abortion tissue, but I don't think they normally do. Let's see, what else? They can get them from cord blood that connects the fetus to the placenta. So there's a number of different sources. It's one of the big, religious people are all freaked out about it for the most part. You know, they think life starts at fertilization, and that therefore a zygote is equivalent to a human being. So, anyway, that's being still worked out. The thing is, they're now getting closer to actually making embryonic stem cells from adult stem cells. So they will probably be able to not have to worry about that anymore. So there's still a lot of promise. Now, one thing that's happened, there are some therapies that they use that have been at least temporarily useful, that work for a little bit of time, like taking the brain cells that control Parkinson's disease and replenishing them. So Parkinson's patients have actually had those cells, they've been losing them. And so they get shaking and they have trouble moving. But it doesn't usually work for all that long, a couple months maybe. At the same time, you've probably heard, like with sports medicine, that they're starting to use stem cells to help athletes who are injured. There seems to be something with that that's useful. At the same time, there's also all these stem cell clinics popping up all over the place. This is something that's going to have to be tackled fairly quickly, because these clinics, they might have someone who knows kind of about stem cells, but they're pretty unregulated. So, for example, in Florida, there was a woman who went in. She's got degenerative or detached retina or something like that, or macular degeneration, something with the retinal cells. So this doctor decided, okay, we're going to try to restore that with stem cells, but he used fat stem cells instead of adult eye stem cells. And of course, she ended up completely blind now. So, just so you know, and you don't go off when you think, oh, maybe stem cell therapy would help, that you just go off to some stupid clinic and go get fixed up. not like that. They've got lots of clinics also in Mexico. They really, there's, the basic science of stem cells has not really gone all that far yet. Oops! All that far, I'm going to need a stem cell transplant. Okay? So I just want you guys to be aware where we're at and be skeptical. Now, we're going to talk about potency. I talked a little bit about this. Not all adult stem cells are the same. In other words, potency is the number of different cell types that stem cells can give rise to. There are organisms that, like us, that have adult stem cells that can give rise to limited number of cells, like cardiomyocytes, which are the muscle in the heart. Those stem cells can give rise to different kinds of heart stem cells. But they can't differentiate into neurons or pancreatic cells or anything like that. Now, embryonic stem cells, as I've been saying, can differentiate into any kind of tissue except for placenta. And so they have what's called greater potency. Okay. Now, there are totipotent cells, and there are pluripotent cells, and there are multipotent, which we'll get to. Totipotent cells from a cell with an embryo with 4 cells can give rise to any kind of tissue, including placental tissue and all the different kinds of tissue in the body. Pluripotent can give rise to any kind of cell. This is from a blastocyst. Any kind of cell except for placental cells. So, this is what I've just said. Mostly the stem cells that we harvest are from the embryos at the blastocyst stage that give rise to pluripotent stem cells. So a single cell embryo can give rise to totipotent. A 5 to 7 day old embryo, the blastocyst, can give rise to pluripotent. And then cord blood stem cells, placental cells are also multipotent. And then there are adult stem cells that are multipotent. They cannot give rise to all cell types, just a limited number for each type of stem cell. But they've been able to reprogram these cells to an earlier, more embryonic-like state, and so those cells can then give rise to a much wider range of cells. Okay. So this is your basic diagram of cell potency. All of these are multipotent. Now, there are still restrictions on embryonic stem cells in research. Way back when Bush was president, since Republicans are really all freaked out about abortion and stem cell research and all that, they put restrictions on they had a certain limited number of lines of stem cells, but they couldn't develop any more. Now that was, Obama came along and unrestricted a bunch of them, but they're still not the kind of thing. In fact, if you're a lab, you have to keep stuff that you're doing with embryonic stem cells separate and separate funding than you get with other kinds of stuff. Now, California decided to fund about $3 billion worth of a year of stem cell research. And so there weren't the kind of restrictions that there were elsewhere. At the same time, remember these therapies could prove vital in making people's lives better. So if someone's a paraplegic and they get a spinal cord injury and repairing that with stem cells, I mean, obviously, they're no longer stuck in a wheelchair. Okay. So, like I mentioned, with Parkinson's disease, it's a kind of a brain cell or a brain cell abnormality. These nerve cells that give rise to dopamine are dying. They've implanted stem cells, brain stem cells, and they've got new nerve cells producing dopamine. Unfortunately, it doesn't last all that long at this point. After a period of time, those cells are also dying and they're not being replaced. So, sources of embryonic stem cells, excess fertilized eggs with IVF, somatic cell nuclear transfer, where they take a cell, take out the nucleus, put in another nucleus, and generate stem cells from that line. Okay, so the nucleus is removed. A new nucleus from a somatic body cell is inserted, and then that cell is allowed to grow, and it produces clones. And then you can do reproductive cloning with that, like they did with Dolly the sheep. And there's a lot of restrictions on reproductive cloning, but in terms of therapeutic cloning, that means trying to generate tissue from stem cells for various uses. That's allowed, and they've been making some fairly decent progress on it. So this is Dolly the sheep. This is the first cloned sheep, or the first cloned animal, using this somatic cell nuclear transfer. Dolly herself was, she didn't live all that long. She lived, normally sheep can live like 30 years, I think, something like that. I think she only lived 6. She had problems. The thing is, they allowed her to breed, and her offspring were fine. So it seems like that first generation of cloning, there are some problems with it. Some people now have cloned their dogs. Some people have cloned prize bulls for, meat production, all that kind of stuff. Now, the thing that's nice about somatic cell nuclear transfer, when we replace body parts from someone else, like a heart transplant, a liver transplant, of course, there's immune response to that. The immune system responds to that foreign tissue just like any other foreign presence and mounts an immune response. So that's why transplant patients get these drugs that suppress the immune system. You have to get it just right, because if it doesn't suppress the immune system enough, it'll start attacking the transplanted cells and you'll get rejection. If you do too much, it means you've suppressed the immune system to the point where you're susceptible to all kinds of infections that your immune system would protect you from. Now, the nice thing about stem cells and somatic cell transfer is you can actually take the nucleus from one of your own cells and put it into an embryonic stem cell after removing the nucleus from that. And you'll end up with cells where you won't suffer rejection from the immune system. Now, eventually we'll probably, well, I'll never see it, but they'll be able to actually grow, let's say, hearts or livers or all kinds of different things from our own cells, and so the rejection problem will disappear. But we're not at that point yet. They have been growing stuff. I think they regrew an ear. They used some scaffolding of some synthetic material and then grew stem cells, used stem cells to grow the tissue for the ear, and then they transplanted that tissue, that ear, onto the patient. That's the outside of the ear, not the actual hearing part.